Dion Annex and Caitlin Mason are the Sherlock Holmes and Dr. Watson of the Lawrence Livermore Lab. But instead of fingerprints and a magnifying glass, they're able to ID their suspects by tracking proteins from their bodies.
What we do is very similar to what is done in DNA, in the sense that there are markers in DNA that you can make a measurement on and then link those to an individual. And we're trying to find the analogous kind of strategy with proteins.
They first perfected the technique with hair and more recently bones. Protein samples are extracted, then run through a powerful device known as a mass spectrometer. It can spot patterns in the proteins that are influenced by mutations in a person's DNA. The result is a unique genetic signature related to DNA, but from a source that's much more durable.
There are many instances where, say, the remains have been out in the environment for a very long time, and the DNA has degraded, or they've been in extremely harsh conditions, say with a fire fatality. In those cases, perhaps the DNA has been destroyed, and maybe the protein has lasted.
Think victims who died in plane crashes, or maybe the intense fires that followed the World Trade Center attacks. The team believes the technique might potentially ID the remains of soldiers from wars of the past and help in cold cases where usable DNA evidence no longer exists. Researcher Caitlin Mason believes the truth is out there.
My heartthrob moment would be exonerating an innocent human from being subjected to being in the box.
In the meantime, the team continues to refine the technique, so one day a fragment of bone might unlock a decades-old mystery, or perhaps the next crime of the century.